contrast, the complexity of life in its simplest form, for it to assemble and to perform, is immense compared to anything else that has been designed, found, or refined, or has come into existence. For example, just to make a molecule that is simple, such as sugar, the molecular machines that life has to make, and the processes that it has to trigger, with such precision and such coordination, the fidelity is beyond our imagination, and the whole system to be able to duplicate so that life can carry on and replicate generation after generation. The chain of events in the fine-tuning, the miniaturization, and the grooming of the aesthetic side stand out and supersede. Only 4 letters, which are then laid down in various combinations, but only in linear fashion in the form of a long chain that goes by the name DNA. These letters are called A, T, C, G. They are short for small molecules that move around free inside living cells. But when many of them combine in a long line, but only in the right sequence, then they are able to spell. The creation of some functional protein, which is a molecular machine that may then be the part of the structure or the function of that living being. Difference in the combination is what makes it possible to code for the creation of proteins of different kinds that we find inside any living cell. But this combination does not happen in random, as DNA can only be copied from a pre-existing DNA template from which the replication machinery can read and build a new DNA chain, following the rule of base pairing, where A pairs with T and G pairs with C, allowing a new complementary strand to form. Life can only become from life. This base pairing is what led to this norm. Protein synthesis is the most important reaction for any species. That is what life has to do. In order to carry out its actions, the machinery and the detailed process is fine-tuned, coordinated, and complex. The proteins are so important. It is not hard to understand, as virtually any medicine you are prescribed, once they are inside your system, they try to interfere or stop. They function of some protein, so the reaction that it controls slows down or halts. Viruses lack the ability to carry out this crucial duty of synthesizing proteins. They lack the machinery. As a result, they have to sneak in to some cell that is still living in order to hijack its tools. For making proteins, detailed instructions are needed. This is what is seated in long molecules, that is the DNA. These long chains behave as life's apps, where instructions are coded and then they are copied and loaded. In large molecular machines, proteins are the final productions made according to these instructions, that we're part of the DNA structure and its function. Proteins are long chains of units called amino acids, which are small molecules. 20 of them exist inside the cell's list, taking various combinations and then laying them down again in a linear fashion is how proteins are made, which amino acids to take, how many and in what order is what the code in DNA dictate. In DNA, the letters are A, T, C, G. Each 3-letter combination is what is known as a codon that codes for one specific amino acid that the cell's decoding machine can read. On top of these, there are 3 combinations that are called stop codons. That tells the system when to end this mission. A specific part of DNA that codes for a specific protein is called the gene. For that particular protein, genes are first copied into mRNA, which has sequences that are complementary to DNA. mRNA is loaded into ribosome, a molecular machine that is the home where proteins are made. It reads the mRNA sequence 3 letters at a time as it passes through its gates and brings the correct amino acid accordingly, and then combines them simply one after the other until it finds a stop codon when it knows that it has finished its mission. A new protein has just been made as the cell continues with this parade. generating random DNA sequence, and then trying to read, you soon find out that you cannot succeed in making any functional protein as you hit stop codons. Pretty soon, so what you end up with is a junk chain of amino acids, unable to perform any function, so the cell will decide to go ahead with the destruction. This finding may be one thing or one objection that goes against any conclusion or any suggestion that the most complex system in its creation, not only of its components but also their coordination and function, including the ability to self-assemble and replicate with such stability, was able to codify all necessary instructions in such a simple and linear structures. Thus, any claim of any superiority by anything else, it very easily ruptures.